we'll just get started in maybe 2 more minutes. And as I mentioned before, please introduce yourself in the chat, and then we'll be using the chat for questions during the talk. Thank you. All right, I think we can go ahead and get started. So it's my pleasure today to introduce Dr. Robert Carnahan for this month's webinar. Rob received his bachelor's degree from Indiana University in Bloomington, after which he spent 3 years working in pharma at Lilly. Following his stint in industry, he enrolled in the PhD program at Vanderbilt, where he remained first as Associate Professor of Cancer Biology and Director of the Antibody and Protein Resource, and now as Associate Professor of Pediatrics and Associate Director of the Vanderbilt Vaccine Center. Rob is a longtime ABRF member, having been involved both in the Antibody Research Group as founder and the Southeast chapter of ABRF, also as founder. I would like to thank Ken Shopman, the ABRF Executive Director, for organizing this event. And also, Rob and I did not go ahead and agree to our clothing choices this morning. But I will say the title of Rob's presentation today is Rapid Discovery of Functional Antibodies for SARS-CoV-2. I'd like to remind everyone to mute their Zoom, and if you have questions, go ahead and put them in the chat. And if you haven't already, go ahead and introduce yourself. But now I'll go ahead and turn this over to Rob. And thanks a lot, Rob. Should be a really great talk.
All right, thanks, David. I'll move over to my presentation here. Okay, can you guys see my screen?
Yes.
Okay, so yeah, thanks. Yeah, I ran a monoclonal antibody facility here at Vanderbilt for about 10 years and was heavily involved in ABRF and benefited greatly from the organization and still a member. And then in 2017 came to the Vanderbilt Vaccine Center to do something a little different. And I'll tell you, the project that we talk about here gets to some of those differences. So here's a little cartoon just to give you, I didn't know what kind of audience we'll have or where everybody's come from. So this is a little cartoon a graduate student in our lab made, sort of the philosophy of the lab. So as opposed to what I did for 10 years using mouse and rat systems to develop monoclonal antibodies, what we do in the Vanderbilt Vaccine Center is focus exclusively on humans. And more specifically, humans who've had viral pathogens by and large. We do a little bit of other work, but mostly focused on viral pathogens. So we will literally look over the world, and we have dedicated personnel to do that, to find people who are convalescent donors, people who have in the past been exposed to a viral pathogen but have completely recovered. And from those people, try to consent them and acquire blood samples that we can begin our B-cell discovery process with. And so here you see those blood samples, and just a little cartoon to show that from those blood samples, we extract the B cells. And then, of course, the B cell people on here would know if you just culture those B cells, they will eventually die. So you have to do something to rescue them, to understand the antibodies that are being produced. And we use two big workflows. One is a hybridomal workflow, which is very similar to the mouse hybridomal workflow that probably a lot of people on this phone call have used. That was the major foundation of this particular group for a long time, but we're now transitioning away from that. It's becoming less and less important, but we still do it. We're now focusing a lot more on what we would call single cell isolation or single cell approaches, which is the entirety of this talk focuses on those. And anyway, the point is we're looking in those patient donor samples for B cells that are making antibodies specific to the target, whatever that target is, in this case, SARS-CoV-2. Fish those B cells out.
Sorry. Made you register again.
Somebody's not muted. Fish those B cells out of the blood and then begin our targeted antibody discovery approaches. And so why are we doing that? And so once we isolate those B cells and start to screen the antibodies, we do invoke this sort of overly grand title, but massively parallel antibody screen, kind of the same that a lot of antibody people do. But what we're looking for here are several different things. What is the diversity of that antibody repertoire contained in that sample? What is the breadth? So, if we were looking at SARS-CoV-2, are we finding antibodies that not only recognize SARS-CoV-2, but do they recognize SARS-1 and MERS, which are related viruses? Down at the bottom there, you see virus neutralization. So we're always looking towards, do the antibodies have a function with specific to the virus life cycle? Can they neutralize? Can they block infection? What are the mechanisms of that protection? So how is it that the antibody is blocking? Is it sort of sterically blocking access to the receptor, or is it blocking the virus on the way out of cells or whatever? And then we collaborate mostly to look at in vivo for protection. So how do these antibodies function in an animal system? So all that to say, you can encapsulate that in this one sentence. We're doing this antibody screening to understand the human antibody response to viral pathogens. And why are we doing that? Well, there's three major things we think about as far as like the practical aspects. So that top part is our basic science aspect, but sort of practical aspects. We're always looking for therapeutic antibodies. Antibodies would be useful in neutralizing the virus. In the middle there, vaccine development. Of course, we are called the Vaccine Center, which is a little bit of a misnomer. We actually don't do vaccines per se. We do collaborate with vaccine developers where we can understand the antibody response. And by understanding the antibody response, you can get a feel for what elicited a good response. And that's sort of what's called reverse vaccinology. So using the antibody response to track your way back to a good vaccine. And then also good monoclonal antibodies are useful in QCing and evaluating vaccine candidates. And then the last thing on the far side there, diagnostic antibodies. Again, we don't operate in that space ourselves. We do make our antibodies available through various mechanisms to people who want to do diagnostic antibody work, and so they can utilize these human antibodies as part of that workflow. So again, thinking big picture, we think of the world, this way we do things in two big ways, at least with respect to speed. We can do this ultra fast in the moment, which is exemplified by the Pandemic Prevention Platform, or P3, which is a DARPA funded program, and we are one of the prime sites for that DARPA program. And that's mostly what I'll talk to you about. We would advocate that that's not the only way one should think about doing antibodies, therapeutic antibodies to viral pathogens. We have another idea called Ahead 100, which is where we're ahead of time trying to find the things that are most likely to cause pandemics and go ahead and make protective antibodies, therapeutic antibodies ahead of time to those. And I'll briefly come back and touch on that at the end. So again, this ultra rapid in the moment is best exemplified by our P3 work funded by DARPA. And so the philosophy or therapy or the idea behind this program was to start with human convalescent donors and to develop a platform one could invoke in the context of a pandemic to, as rapidly as possible, develop therapeutic antibodies. One of the major goals was to actually deliver those antibodies with a nucleic acid where the antibody would actually be expressed in the human, and we're still working on that and have shown some progress there. And you actually see that with vaccines. The Moderna, BioNTech, and other vaccines are already being done in that way. Antibodies are moving in that direction, but they're not there yet. But we are collaborating with people to do that. But this is the point of the DARPA P3 program, is to, as rapidly as possible, develop therapeutic antibodies. And I'll just briefly say that we started this program at the end of 2017. So really 2018, we did a bunch of R&D. 2019, we did our first what's called capabilities demonstration, or sprint, or challenge. And so I've talked about this in the past, maybe even to this group, where we were given a 90-day challenge to develop therapeutic antibodies from scratch, from human blood samples to Zika. I should also mention that our P3 program is not just us. We do the antibody discovery piece. We also have collaborated. This group has changed over time, but at the Zika moment, the RNA delivery platform was the group called IDRI in Seattle. The small animal work was Mike Diamond's lab at Worcester, and the NHP validation was done by Dan Baruch's lab at BIDMC, and we were collaborating with Gleed Alter to do some FC optimization during that particular capabilities demonstration. And so here's again another big conceptual view of what we do. So at the top there you see human survivor PBMC. So that's the blood sample. We get the PBMCs and we start to put them through this funnel. You see in blue there's the PBMCs. In red is one of the critical steps. We're going to enrich for the B cells, which are specific to what we're interested in. And we can do that in a couple different ways. If it's an acute response, then plasmablasts are going to be available that are specific to the target. And we could try to isolate those just with cell surface markers. If we're looking in a more convalescent situation where it's more out further away from the illness, we would look for memory B cells and start to find antigen-specific memory B cells. We would do single cell approaches to separate those all from one another, and I'll talk about a couple different ways we can do that. Uh-oh, my slides are auto-advancing. And then we would use next-gen sequencing to rescue the antibody sequences out of there so we know what they are. We can bioinformatically down select to make sure we have a diverse panel of antibodies. And then we begin the second hard work, which is expressing all those recombinantly and then screening them for function. And then you see there in the red, again, things we generally collaborate with people to do in vivo delivery and validation. So with the Zika sprint, we were given a 90-day challenge. And again, we did that and we were able to do it in 78 days. We did it just as if I call it a fake sprint, but we did consider it like a real sprint. We worked 7 days a week, nights and weekends, across 5 sites in the United States, and we real-time tweeted the whole thing, which was kind of fun. But anyway, that really helped us develop a lot of our capabilities and figure out where the strengths and weaknesses of our platform was. And so towards the end of 2019, we started talking with DARPA again about our sprint, our year 2 challenge sprint, where they are going to challenge us to do the same thing in 60 days, only we're going to have to define a new sprint target. And so that fast forward now to January of this year. Just to remind you of some of the timeline, January 10th, the SARS-CoV-2 genome was published. January 14th, coincidentally, we actually had the DARPA program officers on site at Vanderbilt. And what we were doing was discussing this year 2 challenge. What would the target be? How would we go about it? In that actual meeting, it came up, hey, should we consider this SARS-CoV-2 thing? Maybe that would be kind of like real life, pseudo pandemic to try to respond to. At that point, it was quite small. There were about 40 cases and 1 death. And importantly, there was no known reported human-to-human transmission. And so actually at that meeting, what we decided was, let's not do that. That sounds complicated. All the donors, again, we have to have human donors. All of them were in China. That was, we weren't sure we'd be able to get samples. We do look all over the world for samples, but sometimes that can take 1 to 5 years to bring in samples, depending on the politics and the complexity and the collaborations. So we said, no, let's stick with our original plan. We're going to do a 2020 capabilities demonstration around a target that we're going to define in the next month or so. Well, that didn't last for long because on January 21st, not only did we get the confirmation of human-human transmission by scientists out of China, we also had the first US case was announced. And we got a phone call from our DARPA program officer saying, hey, by the way, I think we're going to change the plan. We actually want you guys to start to orient yourself towards doing all your work for us in 2020 and in 2021 probably on SARS-CoV-2. So we immediately did that. We pivoted around and started looking for that. And on January 24th, just the city of Wuhan was quarantined. Now the case numbers and deaths are climbing. I'm not going to do the typical and go through the whole progression of cases and deaths and all that, but that was sort of a step up in the nature of this pandemic. And then January 26th, we were fortunate enough to be able to get blood from the US patient, which we call Washington 1, arrived at the lab. And that, I just put that out as a testament to the people in our lab who, again, searched the world or searched the country to find these donor samples and connect in with the patient care team and have interactions that hopefully allow patients to allow their blood to be shared with us so that we can do these discovery approaches. I'll just quickly say before we go that we have a really big group here at the Vanderbilt Vaccine Center, but only a small subset of those people were actually involved. You can see in yellow boxes here, they're about, I won't go through them all, but the 2 in the middle there, Seth with glasses next to Jim Crow, my boss, and Pablo next to him, those were really the people, the scientific leads who should get a ton of credit for what we were able to do here. And then a huge amount of technical support by other people, were the people that were invoked in this response, which early on was our entire group was here working during that time while we were responding. But pretty quickly, Nashville got shut down in mid to late February, and is literally, we have a really big space, and it was only these 6 or 8 people who were in that space operating during that time. So I'll just also want to reflect, one of the main things we did during that DARPA visit in January was map out, well, like, how does a pandemic response, like, what have we learned, and what are big conceptual pictures about what you do in response to a pandemic? And we literally mapped this out during that meeting. And we sort of thought, well, the first thing you need to think about when you're responding to a virus is it a novel virus or is it not? Because that would sort of pivot you which way you need to start directing your energy. And so if it's not a novel virus, it's one that we know something about, which has become now evolved into a pandemic. That was best exemplified by what we'd already done, our Zika capabilities demonstration 2019. There, the antigenic targets on the virus were largely known. What you would target for protective antibodies was largely known. So there you have a toolbox, we'll say, which was largely defined, and you just need to implement that toolbox to find the B cells you're interested in, and then have functional screens to find those ones in that pool of binders which have function. If it is a novel or poorly understood virus, well, now life becomes a lot more complicated. One could take a plasmablast approach, and I won't go into the deep immunology here, but that's that first response upon exposure. Within the 7 days, the B cells are expanding, the ones that recognize the target, and those first ones are plasmablasts. That would be great because plasmablasts in themselves can be target specific without having any toolbox to fish them out, because you can just get them because they start to take over the B cell compartments. The problem is they are producing an antibody called IgM, generally, which is generally has specificity issues. It also has production issues. But if you had no other choice, what you could do is respond using these plasmablasts you see here in this diagram, which I won't go through in great detail, but these primary plasmablast response could be utilized and ultimately downstream convert those IgM antibodies over to IgGs so that they could actually be produced recombinantly. So that's one step someone could take, but it would be difficult. That's what we decided at the time. You could also say, no, I'm not going to do plasmablasts. We're going to wait on memory B cells, but probably early on, you're not going to have a convalescent donor available because, again, it takes the body 1 to 3 months to generate these highly affinity matured memory B cells. An alternative is to acquire a donor from a closely related virus. If the antigen was available to allow you to screen for antibodies to that the authentic virus using the donor cells from a related virus, then you could kind of operate on that principle. And I didn't have this in our January talk with DARPA, but just to put it in, actually people did that. Vir Bio, if you go look up what their response was, is they have an antibody which is now in clinical development, which they had developed against SARS-1. So they had a whole panel of SARS-1 antibodies, and their best SARS-1 antibodies actually didn't recognize SARS-2, but they went back into their big panel and rescreened it all using SARS-2 proteins and were able to identify some antibodies from their SARS-1 screen. So they were used this a related virus to actually get to their therapeutic candidates. We did not do that. What we did was largely what you'll see here is we waited. We didn't know how long we'd have to wait. We knew it would be 1 to 3 months. It turns out for coronavirus, it's about 45 days before we could see any donors that would convert over to high affinity IgGs. And then begin your discovery efforts at that point. Hopefully by that point, you have authentic antigen, which we'll cover in a second, and the screening can take place. So if you look back, what did we have? You know, if you see in black, that's what we mapped out when the DARPA program officers were here in January. And our first big step, we actually made a mistake on. We didn't even consider. We thought, well, it's either going to be a new virus or it's not going to be a new virus. And what it turns out we actually had was we had a sort of a new virus, right? It wasn't totally new because it was very, very closely related to SARS-1. But it wasn't SARS-1. It had differences, and we had to develop a toolbox specific to SARS-2. I will say when people ask me, like, wow, this pandemic is terrible, it's like worst case scenario. It's actually not worst case scenario. The worst case scenario would be the yes, right? So we are at the point we're at in the world with both vaccines and antibodies and even therapeutic small molecules because we know so much about SARS-2 because it's so similar to SARS-1. if we hadn't had that leg up where we knew very early on what the target should be for antibodies and vaccines. And we knew that because of all the work people like Ralph Barrick and Mark Dennison had done on SARS-1. So we ended up with a bad but not worst case scenario with this sort of new virus. I will say, I'll go through and talk about our antibody discovery efforts. We're obviously not the only ones who did this. Here's a completely non-exhaustive list of people who've done antibody discovery efforts, a lot of them human, but not all of them, some of them using phage display systems and some mouse work in there, some humanized mice. And so there's a lot out there to read about it, so much that I can't even keep up with it. So coming back to our funnel, so you see down the bottom, this part of the funnel, which is really our part. So I said 78 days, that's to get all the way through animal validation. But for Zika, if you see down in the bottom corner, there's about 35 days for us to do really our major contribution to the work, which is to go from the patient PBMCs on your left to lead antibodies that have been screened on the right. And the first big step is this red box here, enrichment. That's where we have to be able to find those B cells that are making the antibodies of interest. We pretty early on got a plasmablast sample. You'll see a little bit of data from that later on in the talk, where we did see a super high frequency of plasmablasts in a few different donors. But what we also found was they were incredibly low specificity to the target, which is the spike protein, which I'll cover here in a second, on the surface of SARS-CoV-2. So that was a dead end pretty early on. And so then we had to do antigens. We had to rely on antigen-specific memory B cell sorting using flow cytometry. And for that, we had to build a toolbox, because even though SARS-CoV-2 is closely related to SARS-1, it is still distinct in that spike protein. And we had to actually produce that spike protein to be able to fish out those antibodies. So again, SARS-1 to the rescue. Fortunately, so much known about SARS-1, we are all, as a world, pretty quickly able to identify that SARS-CoV-2 has a protein on its surface called spike. You see here these little blue knobs on the surface. And spike protein, at least one receptor, this is starting to change, but one receptor we know for the virus accessing human cells is via spike protein binding to a protein called ACE2 on human cells, expressed on a lot of different cell types, it turns out, but mostly on endothelial cells. So spike binds to ACE2, and then through that sort of gets endocytosed, and that's how it infects cells. So our first job was to build the toolbox around these two parts of the puzzle to be able to identify the antibodies. So the antigen, which you see here in the center, is the spike protein, which forms a trimer. And thankfully, a bunch of different structural biologists, but Jason McClellan's lab at University of Texas was the first to publish not only the structure, the cryo-EM, but also the method to make soluble full-length spike trimer protein, which is what we used throughout the entire sprint time. We also made the subdomain of that protein called RBD. That's the receptor binding domain. That's the piece of spike which binds directly to the ACE2 receptor. And then we're also ramping up in our toolbox and making recombinant human ACE2, the soluble ectodomain of that as well, because we needed all those to be able to not only fish out the B cells, but to be able to do functional assays on those B cells to see if we could find antibodies. Again, now you can already see what the goal is going to be. Can we find antibodies that interfere between the interaction between spike and ACE2? And can we disrupt that interaction so that the virus cannot access the cell through the ACE2 receptor? I'll just introduce, because along the way, to put it in context, Nashville was locked down during this time. And if you've ever been to Nashville in the spring, David knows we have really rainy, dreary springs. Nashville's a beautiful place most of the year, but the spring we get a lot of rain. It was very dreary. We were on lockdown. We were one of the few places working. And again, we have this huge center where only 6 or 8 of us are working. We spent January and most of March making tools, which turned, at least like spike protein and tools, which turned out to be way, way more difficult to make than we hoped. And we were processing donor after donor, which had a non-optimal responses. So one of our ways of coping, like a lot of places, was gallows humor. And so we started this meme door, which I'll come back to a few times, just to kind of give you a feel where we were in our feelings and in our thoughts during this time. So one of the first ones is this, the spike protein, which again was critical for us to make to be able to fish out those B cells, was really difficult to make. We make a lot of viral surface proteins. Spike turns out to be one of the lowest yield ones we've ever made. And so you see this, this meme someone found here. ICOR is the group of people that were making the protein, Rachel Sutton and Rachel Nargi. And here's it ain't much, but it's honest work. So during the entirety of this 2 or 3 month sprint, we literally had spike protein going and harvesting about every 2 days. And we would use that up, and 2 days later, we would make another couple, 50 micrograms, 100 micrograms, and we would use that up. And that just went on for month after month during the entire spring. I won't go through with this. This is from one of our papers where we're kind of mapping out, well, what did we do with those tools once we had the spike protein? How were we going about using it to do our B cell discovery work? Well, we had two major workflows. This is a really busy slide. I probably shouldn't use it. But in the red box on the left, you see discovery approach 1, which is a single cell sequencing approach, which is one that we've been using for several years based on 10x chromium device, which I'll cover briefly. And the one on the right was a single cell functional assay approach using a beacon from Berkeley Lights, which we had never used. Honestly, they called us after we started responding and said, hey, we want to collaborate with you. What if we came there and we worked together, which they did. They sent people here and they literally were working with us during a good part of the early part of the sprint to do a completely different and parallel approach for identifying these B cells of interest. But at the end, if you went and looked at this figure, they basically converge. Those are just the discovery. How are we fishing out the antibodies of interest and getting the sequence to those? But ultimately, what we need to do is express those recombinantly, which you see here in the very center of the slide, do an expression, and then start to characterize those either by antigen reactivity. You see on the left, like, are we binding target? And then on the right, is that binding of target, is that functional? Is that able to inhibit virus infection or egress or whatever? So I'll cover, again, these discovery approaches in just a bit more detail. So the front end of them is the same. You see in red there, we isolated and enriched B cells using flow cytometry, again, using probes made from spike protein. Throughout the entire process, we were using both soluble trimer spike, and we were also using the RBD domain. And so that we're putting on the B cells, it's finding the B cell receptor, and it's labeled so that we can use flow cytometry to sort out those cells which are making B cells specific to the target. And then the beacon approach, the important thing here is it's functionally specific from the beginning. You're actually using those primary B cells and screening the antibodies they're making, and then only following up on those B cells making antibodies that are interesting. But it's a lower throughput approach. And so here you see a little cartoons of it on the left. We do antigen labeling, like I said, using full-length spike or RBD. So those were labeled, and we're sort of fishing out and lighting up the B cells of interest using flow cytometry. We then expand those in cell culture, and then we load them onto the chips, they're called, on the beacon. And here's a little picture of how the beacon works. I won't go into the diagram again, but we'll start it in B up there in the top left. What happens is, basically, this uses microfluidics to spread these B cells, which are loading onto the chips, such that there's about 1 B cell per well. They call them a pin, a nanopin. And so, and then you can do, so if you look here in like the second pin, you can see a light down there. That's a B cell down there. And then they secrete antibodies, which come up the pin, and then the antibodies blooming out the top. And you can do assays in two ways. You can flow reagents across the channel, it's called, and look for signal there, or you can actually do assays down in the well, and we did both. And so here, what you can see in this particular one is we're looking for antibodies to the spike protein in the top right. And we're doing an in-channel assay. So all of these pins actually have a B cell in them, even though you can't see it because we don't have a bright field image, but there's a B cell in the bottom of each of these pins secreting antibody. And you can see there are 3 pins which are secreting an antibody that is binding to and causing the secondary agent to bind to and light up in the channel above them in this bloom. So then these are now identified as the B cells in these wells are making spike-specific proteins. We also could, in a different color channel, float in that same channel a reagent beads that have just the RBD domain. And what you can see here is that there's a well here that's also specific to RBD, right? 1069 is making a protein, making an antibody which is specific to spike and RBD. So that's the subdomain of spike. We already know that the antibodies from this one are RBD specific. And so that was one of the assays we used, basically purely a binding assay using single primary B cells and studying the antibodies they're making. Here's a little video where you can see these blooms forming. Just a second. So you can see above many of these pins, they're lighting up as the antibody is being secreted from the B cell down at the bottom and getting into the channel, and then lighting up these reagents which are floating across the top, these these beads, and then the beads are getting labeled and lit up in the appropriate channels. And so this gets us to a point where we're able to, again, identify both spike-specific protein antibodies, and then a subgroup of those which are binding the receptor binding domain. So here you see we have 3 B cells making spike-specific, but only one of those is making an antibody, which is binding the RBD domain. I'll just insert here. I won't go through this whole flow cytometry, but just because on the next slide, you'll see we used the beacon to characterize not only the memory B cell response, but the plasmablast response. So we went through this fairly complicated process to fish out plasmablasts using cell surface markers from acutely infected donors. And we were able to very successfully fish out huge populations of plasmablasts, which you just kind of can see. Just take my word for it if you don't follow all this, because honestly, I don't either. This is done by the technical people in the lab. But at the bottom right, we're able to successfully isolate plasmablasts. But what you see here from our beacon picture is on the left, now we're looking for all of these pins have B cells from that plasmablast enrichment, but only 2 on an entire chip, which is hundreds and hundreds of B cells, are making antibodies specific to spike, and none of them were specific to the RBD domain of spike, which we already knew was our principal target based on SARS-1 work. We knew the RBD domain was what was going to dock into ACE2. So pretty early on, these plasmablast samples were not super interesting to us, especially when we've got, you see, this is a month later, so we have our antigen sorted memory B cells, where about 55% of the sorted from the flow cytometry B cells are making antibodies that are specific to SARS-CoV-2 spike. And you can see that here by all the blooms on top of the pins where there are B cells. So that's the binding assay. We also were able to mount onto the beacon a functional assay where we would look for the ability of the antibody to interfere with ACE2 spike interaction, where those, so here you see this this well is making an antibody which is RBD specific. You see the bloom at the top of the very first sort of box there. We also had RBD reagent bead down in the well, and you could see that the B cell, which you can't see, is making antibodies which are binding to this RBD coated bead, and then secondary reagents are lighting up, so it's RBD binding here. And then we have ACE2 unlabeled in there. No, ACE2 is labeled as well, and you can see that the ACE2 is able to bind to that bead as well, because the bead has RBD on it, and so it's binding both antibody and ACE2. So this is a non-blocking antibody. But in certain instances, you can see where we have, now again, we have an RBD binding antibody. You see in the channel assay, it's making a bloom, so it's RBD specific. It's binding RBD coated beads down in the bottom of the pin. And now when we try to have ACE2, fluorescent ACE2 bind those RBD beads, coated beads, it's not able to, because the antibody is there blocking ACE2. So this was one way we identified functional antibodies. And I'm just going to skip forward because I'm kind of feel like I'm running out of time already. But we can come back to that if people want to at the end. The other was this 10x chromium approach. So this is functionally agnostic. We don't have a functional assay on the front end. Here we're just going to do that B cell sorting using flow cytometry, find the antibodies that have making an antibody specific to the target, and then single cell encapsulate those and use next-gen sequencing to sequence all of them. And 10x is great in that it'll help us keep the heavy chains and light chains together so that they're associated with one another. We know which heavy chain goes with which light chain. And then we use bioinformatics to sort through. That'll give us thousands and thousands of antibody sequences. We will try to use bioinformatics to down select and get that number down under 1,000. And then we will just synthesize and express all of those. So we did that in this approach and ended up identifying about 1,000 antibody sequences that we expressed in parallel, in addition to what was coming off the beacon, which was a few hundred beyond in addition to that. And this is just a diagram showing that, I won't go into detail about that. So now we come to the timeline, February 2020. Again, this is when we were doing these plasmablast samples. This is from our meme board. We were all getting pretty tired at this point and getting kind of gun shy about the samples which were showing up and continually showing us that they did not have antibodies contained in those donor samples that looked like they were worth throwing the full weight of our energy into. But lo and behold, March rolls around and we identify two of the earliest donors and two of the earliest infected patients in the North America. They're actually in Toronto. We were able to work with local people, their physicians, and the donors to be able to acquire samples from them. those actually got lost in the Toronto airport for a while, and FedEx was sent, literally sent like their special ops team into the Toronto airport to find them and rescue them for us. And they arrived here on March 14th. And now I'll talk to you a little bit about what happened after that. I won't go into the details here, but ultimately led to a panel of about 1,000 antibodies, which we went on from, and all really came from those 2 samples that came from, the best of which all came from these, the best of our antibodies all came from these 2 donor samples, which came from Canada in mid-March. So once we had done that discovery approach, which I already described to you, both the beacon and the 10x, we have again these 1,000 antibodies we need to express. We used our microscale format, which is honestly not super technical. It's just having optimized 96-well plate expression of all these in parallel, but we've worked with it enough to be able to get about 40 micrograms on average from a 96-well plate. 100 microliter culture of XP CHO cells gives us about 40 micrograms, which is really all we need. We've downsized all of our screens to be able to do everything we need to do initially with that 40 micrograms. And of the 900 antibodies, we were able to successfully get about 760 of them to express well. And then we went into this, and so then we kind of go back. This is just a different view of our overall process. At the top, again, you see the antibody sequence rescue, which we kind of do in the discovery approach. We do microscale production, which from 100 microliter gives us about 40 micrograms. We can do hundreds to thousands of samples. It's about a 5-day turnaround to get that antibody to start screening. We do the screening, we find hits. We then go to what's called MIDI scale. Now we're at 15 mLs of culture XP CHO. We get about a mg of each one. We can do 10 to 100 of those in about 7 days. And then lastly, far on down the line, now we're already into like May, we can do maxi scale production for NHP validation, which we did with our 2 best antibodies from this sprint. And there we were using about 5 or 6 liters to get gram quantities. We do all that in-house. So we can do 2 or 3 samples of that. And it takes about 14-day turnaround from the transfection date to having pure qualified protein in hand. And I won't talk a lot about it, but particularly the microscale and midi-scale, have hugely benefited from this instrument called the Protein Maker, which is 24 parallel purification sort of tracks that can be all operated independently. And it's really helped us to be able to do these super high throughput protein purifications. What came out of that was most of our neutralizing antibodies mapped to the RBD. 67 of the 70 were able to find neutralizing activity. This diagram on top kind of shows you the specificity in blue of where they're mapping onto whether it's RBD, S. Each line is a different antibody. So you can see most of our antibodies were to RBD, but we did have some other ones. But on the bottom, you see where the full or partial neutralization, a lot of that's mapping to the ones that are RBD specific. How do we do this functional screening? We use this device, which we'd been using for a couple of years in developing this way to do viral neutralization, which was very different than the traditional plaque assays, if you know what those are. Basically, what we use to do these viral neutralization screens is based on the principle of cellular impedance. So we use microwell plates in a device made by ASEA, and on the bottom of each well are 2 gold electrodes. And electricity is continually flowing from one electrode to the other, right? So if you just have it going, it'll detect that electricity is flowing. If you put cells into the well, they will settle down onto the bottom of the well and begin to impede that signal. So that can be read out like you can watch it in real time, see a trace. The impedance is going up as the monolayer is being formed. And so you can, here is a trace where you can see that. And so in all these lines, the cells are added at time 0. And you can see the impedance is going up and eventually plateaus. At that point, you can add virus, and you see in gray what would happen if you just added virus alone. You would have some period of time where the cells are being infected, and then ultimately the virus will start to cause what's called CPE, cytopathic effect, and in many cases called cell lysis, and the cell monolayer will start to crash, and the impedance goes back down, kind of returns to baseline. So that's in the gray line. Sorry, my slides keep auto-advancing. I'm not sure why. And if you put in a protective antibody, if you put in no virus, then you can see that in blue, right, where it would just stay as a monolayer. And then now we have in green a fully neutralizing antibody. So at this point where you add virus, we also add a protective antibody. In the green line, you can see what the antibody does is protect the monolayer as well. And then you can have partially protective ones where the antibody protects to some extent. So it's better than no protection in the gray line, but the red line shows you sort of an intermediately protective antibody. So we use this, and doing this, standard virus assays where you're looking for functional neutralization, those can take 4 days to 14 days, depending on how the system exactly works. We can actually, as you can see here, we can get our first readout. We definitely know by 60, 70 hours the final result. But you can see here, if you look around 36 hours was an inflection point where we pretty much know what's going to happen at 36 hours. So we have a 36-hour assay where we can make a call whether an antibody is protective, yes or no. And here's just some more diagrams of showing it, but for interest of time, I'll just skip this slide. And I will say, because this was a BSL-3 virus, we actually had to transfer this technology mid-sprint to some of our friends in the Diamond Lab at Washington University in St. Louis and teach them by phone how to do it, because they had to move it into their BSL-3. And we were literally doing these virus neutralization assays at certain points with them. One of them outside the BSL-3 getting instructions from our scientists, and then going into the BSL-3 and telling them, well, here's how you need to load the machine. And they actually had the computer. They had someone come in and hook up the computer so we could all watch it live, even though the computer was in the BSL-3. But they did a fantastic job. And here are Brett Case and Swathi, who actually did all those assays. And then they confirmed it with traditional virus assays. And so you can see here on the far right, a big panel of antibodies that we got, all of which were the binding antibodies. You see S2P binding, that's full length spike, and RBD binding. So these are all of our binders. But what you can see is binding doesn't mean blocking. That's our functional blocking assay, and definitely does not equal neutralization. So you could have a whole range of neutralization values, even if an antibody was quite good at binding. And so here's our next meme board that popped up after we started getting our results. We got potent neutralizing antibodies and everybody was happy. And we started to sort through those more deeply now, doing animal work, which again was with our friends at Washington University in St. Louis. And so I guess I'll come back to that. I don't know if I should cover this. There were basically what we defined, the RBD domain, which turned out to be the domain where we could track neutralization to, is a very small region. And what we have essentially decided, there are multiple sort of ways antibodies can bind it, but there are 3 principal sites at which the antibodies can bind. One, which is, you see here, this is a blocking assay. So in black, these antibodies all block one another, indicating they bind the same epitope. And then there was a second site, which was non-neutralizing. And then a third site, which was also neutralizing, but not quite as potently as the first. I won't go into big detail about that. But what we ended up doing was trying to build antibody cocktails that bound to RBD non-competitively. And we identified a cocktail, 2196 and 2130, that bound very well together. In fact, what we ultimately defined was that they synergistically bind. The binding of one of those, which I won't go into the math behind this, but we have a super genius guy in this lab who figured out that that the binding of either one of these antibodies helped increase the binding and neutralization of the other. So they were synergistic, not just additive. And that became sort of the key cocktail that we moved forward with in our animal studies, which again, for the sake of time, I won't go into grave detail about. But working with Mike Diamond's lab, we also work with Ralph Barrick's lab in another animal model. So what's happening here, this was an older model, which is now kind of fallen by the wayside now that people have the transgenic ACE2. The thing to know is that mouse ACE2 is not infected by SARS-2, so you have to add in human ACE2 in some way. And so in this case, what Mike's lab was doing was adding it in using adenovirus, and they would add in the human ACE2, and then they would infect after that. And what you see is in black, if there's just an isotype control, the animals lose a lot of body weight and eventually have to be sacrificed. But with protective antibodies, either individually or as a cocktail, those were protective, not only for weight loss, but you see on the far right, pathology also showed that if you just use an empty adenovirus, the cells look nice. Isotype control, you see a lot of immune cell infiltrate and disruption of the endothelial cells in the lung. But with 2196 or any of the other monotherapies or combination therapies, we could block that phenotype that was seen in the lungs. Once we down selected to a couple lead candidates, we have another collaborator who's part of our DARPA program, Dan Baruch's lab at BIDMC, who was able to rapidly do non-human primate studies in protection. So these are protection, not therapy. So the antibody was given prior to viral challenge. And what you can see in the isotype controls, we'll start there, after viral challenge in the nose and in bronchiolar lavage, which is telling you what's going on in the lungs, you see a rapid increase in viral load in both the nose and the lungs, which is completely prevented if you pre-treat the monkeys with either of these two antibodies we were able to study in this particular study. And so just to now recap what happened, the sample arrived March 14th. On April 8th, we were able to transfer the sequences to several pharma partners, one in particular, and then we continued to do studies and collaborate with them beyond that point. So we're able to do our antibody discovery effort, which was 35 days for Zika. We were able to cut down 10 days and do it in 25 days, which was an achievement. It was not without some suffering. So here's some of the memes that were popping up around the time where it seemed like the sprint was never going to end. You see Elmo becoming animal and Tom Hanks looking a little worse for the wear here at the end of the sprint. So that's kind of how we're all feeling because, yeah, during the time it was pretty intense. I'm sure other people working on it probably know that. We actually set up beds. Our administrators weren't allowed to come in, so we took the administrative offices and bought a couple beds on Amazon, a little fold-out beds and put in there so we could have the people who needed to sleep here could sleep here when they needed to be. I'm not sure I'm supposed to say that because our HR department may not like that, but that's what happened. So we feel pretty good about how it ended up. So one of our goals, again, is always to do therapeutic antibodies. And we pretty early on, even before we discovered the antibodies, started working with AstraZeneca and having lab meetings with them. And they ended up picking up several of our antibodies and are moving forward with the combination of two of them, which have completed phase 1 clinical trials, with phase 3 is already enrolling, I think, and to begin any day. We also worked with diagnostic companies, some of which the antibodies are being incorporated into platforms. And we are working with several vaccine manufacturers to use some of these antibodies for lot release in clinical studies. Whew. So that was a whirlwind. I wanted to kind of get through it, but that's our ultra fast. I won't talk about it, but I would say we don't know that that's the way that we should be doing it as a society or as a science. We would go with Ben Franklin's idea that an ounce of prevention is worth a pound of cure. And though we may not know exactly the pandemic targets that are going to happen, this is actually SARS-2 is a good example of how much we've benefited by having worked on SARS-1 and having therapeutic antibodies and drugs to SARS-1. And we should continue to do that through all these viral families. And we call that a Head 100, where we're just continually searching for the next possible pandemics and going ahead and making antibody therapeutics and drugs to those ahead of time. And I did it in still 12 minutes. So as we say here, good science is team science. So this is the full vaccine center, which is a pretty big group. And again, right there in the middle, a lot of credit to Seth Zost and Pavlo Gilchuk, who are the lead authors on our papers that came out. And we're really the brains and the energy and the passion behind everything we were able to do here. So with that, I'm sorry if that was super fast, but it was a lot to cover. And I wanted to kind of put some of the context in there, not just the science, but I am happy to take questions, and I will stop sharing my screen.
All right, well, thank you, Rob. That was really great. So I just want to remind everyone, if you have questions, the way we're going to do this is put your questions in the chat, just so we don't have people talking over each other. And then I guess Rob can read the questions, and we'll repeat them and provide some answers. So I'll go ahead and start it off. So I put a couple of questions in there. One was about this beacon. So Berkeley Lights, I've heard a bit about it. It's a bit expensive equipment. So, I mean, you guys are pretty lucky that you got one, but can you just describe that system a little bit? You know, how many channels and kind of a little bit about it?
Yeah, it is very expensive. Again, we were able to we developed a collaboration with them. They mostly work with pharmaceutical partners who can afford very, very expensive equipment. Actually, the beacon has made most of its claim to fame through cell line development, where it can do super high throughput cell line development for CHO cell manufacturing cell lines. But we use it for antibody discovery. Sorry, how many pins, I think how many sort of pins? It depends. They have channels that do all kinds of different levels of pins. I think the one that has the most is 17,000 pins is their biggest chip. So you can load in about 17,000 B cells and screen them at the same time. You know, it's like everything, not only is the instrument expensive, like a lot of these instruments, the consumables, the chips are one-time use. So you have to kind of put those into the process as well. As far as the antibody discovery timeline, I see that's your other question. We get that question a lot. We also get that question from the technology we're using. You're like, who's faster, us or them? And what we sort of say, it ended up being about the same. You just get a different result, right? So the beacon, yeah, you get the sequence faster. You get the antibody. Well, you actually get the B cell faster, but you have to clone it yourself. And whereas with our 10x approach, we just get the sequence and we send that out to a third party to synthesize and clone it for us. So it's sort of speed, but there's also how much work you're doing yourself, right? So with the 10x approach, we're just waiting for a couple of weeks for the synthesis to come in. We can start to prepare reagents, whereas the beacon, it's constant. You're working, we're doing all the PCR ourselves, and then sequence validation. So they end up being about the same, really. They're both, the discovery parts is, several weeks if you're going at breakneck speed.
All right, I guess if you want to go in and read down the line there, we've got one from Francis. And
so what lessons have you learned? What would you do differently with the next target? Well, we would learn that the lessons we already knew, but we didn't pay attention to, which is we kept doing donor after donor in those early days, knowing that it was probably a waste of time, but feeling so much pressure to do something. But it was all a waste of energy. The human immune system, we kind of know the timeline of the human immune system. We kind of knew trying to find donors in the first month was a waste of time, but our sponsors and other people really wanted us to be doing something. So we were just working 7 days a week for processing samples just to be able to, like that Washington 1 sample that we spent all this energy to get on January 26th. Nothing came out of that. No good antibodies came out. It was too early, right? There's nothing good going to come out of that. That's one. The other is we need a coordinated national response. We had to go source every one of these donors ourselves across country lines. It was super difficult. We actually were tied in very early on, like within a day or two of the Princess cruise, we were piped right into the people controlling those patients and had every access point possible to them. But bureaucracy intervened. And we were trying to talk with them about these people were willing to donate, but there was bureaucracy between us and getting samples. And so much bureaucracy that we ended up going a different direction. And the person in control of those patients wrote us this was like in February. And they wrote us in the beginning of May and they said, hey, good news. We have all the paperwork done. We can now give you samples from all these donors. And we're like, hey, good news. We just handed off all of our antibodies to AstraZeneca. I'm thankful that you can offer them, but we don't need them anymore, That happened over and over in different ways. There was just no way for someone to break the glass and pull the fire alarm and say, I understand IRB has a reason and consenting has a reason, but there has to be a way to make it faster. Like we can't do the normal. So that was the lesson. I hope that answered your question. So someone asked, what can you tell us about the manufacturability of antibodies? Are other modifications needed? That is a big deal. I will say that was one of the huge benefits of us. One thing we did a little differently, I think, than some of the academics is we were very fortunate to be part of DARPA. And so we were working with pharmaceutical partners in the midst of our discovery approach, not at the end. Like they knew our sequences and they knew what we were doing while we were discovering it. So they have very good analytical tools for manufacturability. And so our job was to make big, broad panels and for them to help us down select those. We have our own manufacturing analysis tools as well, but that was kind of nice to see our analysis tools and their analysis tools and see kind of like, if you look good in both, then you're probably going to be good. And that turned out to be the case. But it is a big deal. It's a less of a big deal with human antibodies, I will say, than with mouse ones, because we don't have to worry about any of the, converting over to human antibody stuff, which we've probably all done at some point, and has a lot of complexity to it. These were human from start. And so they're being made in CHO cells and they tend to behave well. Most of them are manufacturable without too much problem. And the only modifications needed are kind of like, depends on what the companies want to do. Do they want to add a long half-life, which is a mutation you can add in the constant region to sort of take the normal antibodies injected into human are going to circulate for about 30 days and then be removed from circulation. You can make mutations that interaction with the receptor that pulls them out of circulation, which is what AstraZeneca and other people have done that give them more like a 90-day half-life. That's one set of mutations. And then we can talk about, I won't go into effector function, You can make mutations to block interaction of the antibody with effector function, which is going to be interesting. We're conducting a worldwide experiment. No one has really been able to know, like, is it good to completely eliminate effector function for viral antibodies, which is the camp we're in. In the vaccine center, we say, you don't need effector. You just want to block virus. But there's a whole nother camp that says, no, 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 no. You got to have effector function. We think that's dangerous. It could be complicated. So they leave the antibody just like it is. So you'll not only get binding neutralization, you'll get effector function happening, other cells interacting with it, and you'll get whatever comes out of that. So that was, again, a long answer. David, can you remind me if the 10X is hybridomas or EBV treated? The 10X is not hybridoma. We don't do, it's where we're taking, expanded, we sort the B cells, we expand them actually on a functional cell situation. We don't actually use EBV for that, but we have a related technology to allow the B cells to expand. And once they're expanded, then we'll single cell encapsulate them. So each B cell is encapsulated. They get barcoded so you know which heavy chain gene goes with which light chain gene. So that's the 10X technology. And what parts of this work should be included in the undergraduate biotechnology course? Honestly, it's so funny. I'm super proud of our recombinant antibody production. Like people, like the two or three people who've worked on that. I mean, getting 40 micrograms from a single well of a 96 well plate is amazing. And they spent a lot of time. I think for a biotechnology course, it's like a testament to like carefulness and doing the right experiments and low tech. There's no fancy machinery involved. It's just figuring out how to do it, like the volumes and playing around with it and how you're going to shake it so you get good aeration, and all these things. Because these cells are obligately suspension cells. So I think those experiments are super cool. And then also the MIDI scale, which we figured out during the sprint, we had already developed the microscale where we could get tens of micrograms from a 96 well plate. And we knew we could get grams when we had to, but we also knew we needed to figure out something in the middle that wasn't just doing 10 wells of a microplate, because that wasn't going to work. And so people were able to play around with some different versions of 50 mil conical mini bioreactors and be able to get to a point where you, again, figuring out aeration and volume and purification techniques to be able to get, a milligram of antibody from a 15 mil culture. I think that's super cool for biotechnology, but I don't know, maybe other people think that's too narrow.
Right. So, we got one more question coming in.
Yeah. Is the mini and MIDI scale published? Definitely the mini is. So we had an article, we took the Zika sprint. We actually had that paper published in Nature BME earlier this year, biomedical engineering. And in there is a pretty detailed description of how we do the mini scale. The MIDI scale, we didn't have at that time, but I think it's in the SARS. If it's not, it should be in one of the SARS-CoV-2. We have 2 SARS-CoV-2 papers out from our lab. If you don't find it there, let me know. I'm happy to share it. Again, it's not super fancy technology. It's just shakers and smart science people who figured it out. I'm not one of them, but I'm super proud of what they did. Yeah, and so how about parallelization? We try to keep, so it's funny, people focus a lot on the beacon and the 10X and the fancy instruments, but we do try to keep it simple where we can. So all of our screens are just straight up old school ELISAs, man, just well done ELISAs in 96 well plates. So we can screen binding in parallel. Exactly, woohoo. We try to do the microscale plate expression just in 96 well plates, and then put in fancy instruments like the protein maker. I will say that's an example of an instrument which was so critical to be able to do 24 independent parallel expressions. Transformed how much work it takes us to do the prep. But we like everybody else, we've suffered from the shiny light and getting the big fancy instrument, and sometimes it works and sometimes it doesn't. But so the protein maker was an example of like that was an awesome addition to our toolbox. But try to keep things simple where you can. Yeah.
All right, great. Well, thank you, Rob. That was really fantastic talk, and we appreciate your time. I know you guys are probably still right in the thick of it, working on the next targets. But yeah, thank you for a great talk. I
Yeah.
will virtual applause indeed. So I'd like to remind everyone we're going to continue this theme of SARS-CoV-2 and antibodies. So next month we're going to have Dr. Breanne Barker. So she's a host on a pretty popular podcast. It's become more popular now called TWIV. And so Breanne will be joining us. We're really excited about her. We are starting to fill out our schedule for next year. So if you're interested in giving a talk, if you have something that you'd like to talk about to our antibody group, contact me, and I'll go ahead and put my contact info in the chat here. We're really grateful to all of our guest speakers, but it's really a community here. So if you have other things you like to talk about, or you know someone, please let us know. And then finally, I'd like to thank Ken Shopman and ABRF for providing this platform. I know for Rob and myself, we have been involved with ABRF for a long time. It's a great community. If you're not familiar with them, check them out and become a member. Can't have too many great things to say about them. So
Get involved. It's great.
yeah, definitely get involved. All right. Well, thank you for your time, and we'll hopefully see you next month on November 16th.
All right.
Hey,
Thanks,
take care.
everybody. Bye.
Bye.